No, like this, patients are fine. Oh, they always.
Okay, let's get going.
Yes,
Okay, we talked about the pancreas, pancreatic cells, alpha, delta, beta, insulin, the main hormone regulating blood glucose. Important to maintain glucose for the brain. We couldn't get the lights balanced.
No.
What happens when we turn them off?
When you turn one switch off, they both go off, and when you turn the other switch off, it goes on more.
Oh, weird.
Can you see enough to write like this?
Yeah. Oh, okay. I didn't know.
Give it a second, your eyes will adjust. Okay, so going on. Insulin action. What does insulin do that gets glucose into the cells? It inserts the GLUT4 molecules into the cell walls.
Hold on. Wait, let's.
Acts to remove excess glucose from the blood after carbohydrate-rich meal. It inserts GLUT4 carrier proteins. This is something they did not have a clue about when I took my first physiology class. They didn't know about these. They figured, like the proteins, the aquaporins were always in one spot. What they found out is they move to the parts of the cell that need them. When insulin, the action of insulin inserts the GLUT4 into the membranes, allows the glucose into the cells. Okay? Promotes production and storage of glycogen. Fat inhibits the breakdown of fat and muscle. If you've got insulin produced because there's a lot of carbohydrate, you're not going to break down fat and muscle. You're going to store it. Skeletal muscles for insulin. Intake glucose for energy, responsible for insulin stimulating glucose uptake. They can also take in glucose and convert it to glycogen for later use. We already talked about that. Only skeletal muscles make glycogen. They cannot use it for their... Once skeletal muscles make glycogen, they must use it for their own energy needs. Once a skeletal muscle has glycogen, it's in the skeletal muscle, cannot go back into the blood.
So, but doesn't the liver make glycogen too?
Yes, and the liver can release it into the blood.
Okay.
Glucose taken into the liver and skeletal muscles converted into glycogen for storage.
Gotcha.
Limited room. Once that's filled, the excess is used as fat. Why don't we just store it as glucose?
Because fat holds more energy than glucose does.
Because it probably takes longer to break down fat.
We can't.
It's a room thing. It's a size volume thing.
Fat takes longer to break down.
It's an osmotic thing.
See, we can't.
It's an osmotic thing. Remember, each molecule, if the molecule's this small, it exerts one molecule of osmotic pressure. If the one molecule is this big, it still only exerts one.
Wow.
So if you have a billion small ones, they're going to create an osmotic pressure in the cells and in the muscles. It's going to draw so much fluid in that it's going to destroy it. That's why you produce glycogen. You only store so much glycogen, you do it to fat. And fat is not active, if you know what I mean. Lipids are not
They're non-polar. They don't like
It's
playing
been
with
a
nobody.
long day. You see what I'm saying?
Yes.
No.
Yes.
Okay, well, then you didn't see what I'm saying.
That's
Good
good answer.
Increased blood glucose enters the cells, closes potassium channels. I don't think you're going to get into this much of it.
Okay.
Regulation of insulin secretion.
Ooh, that's a good picture. That'll be on my cheat sheet.
That's going on mine.
Regulating metabolism, adipose,
How insulin is
liver tissues for sugars, hormonal regulation of metabolism, insulin and glucagon diabetes.
regulation of insulin secretion.
After carbohydrate-rich meal, increased circulating glucose inhibits alpha cells, So glucagon is not released. It activates the beta cells. Promotes. Let's see where we're going.
The slides of these words.
Mine.
Are they from the text?
Some of them are from the text. They're from the slide. It's from the instructor's part of the text. Regulation of insulin. Hang on. Insulin acts on the hypothalamus to inhibit conversion of glycogen to glucose in the liver. Okay. It inhibits the release of glucose from the liver. Insulin.
Yes. Oh, see, I asked Patrick that. I asked him, does insulin will, as it, uptakes, it inhibits, and he goes, no.
It inhibits.
It does inhibit the glycogen.
When insulin levels drop, this inhibition is lifted.
Yeah.
And then the liver starts putting sugar out into the blood. Okay?
That's what I thought.
Increased blood glucose, enter the beta cell, close potassium channels, and that the calcium releases, calcium stimulates the insulin release. Opens the calcium channels, stimulates insulin release. So take-home message is the higher blood sugar, the more insulin you're going to have.
That's right.
Okay?
Higher blood sugar, higher insulin.
Yep. And then when the blood sugar goes down, the insulin goes down. Regulation of glucagon after carbohydrate-rich meal. Glucagon is not released. Glucagon is released from the alpha cells in order to free up sugars. If you don't need the sugar, you don't need the glucagon. When the glucose levels falls, the inhibition is lifted and glucagon again is released. Glucagon prevents the cellular uptake of glucose, promotes conversion of glycogen to glucose, and releases glucose from the liver.
Is this what we talked about SI with sympathetic and parasympathetic, I think?
Yes?
Do you remember that with
Yeah.
Yeah. When you're fight or flight, your body's doing this to release as much glucose as it can
Right.
quickly into the bloodstream.
The insulin.
Yeah.
But it does that with epinephrine, not glucagon.
Epinephrine can
Yeah,
act like
but-
glucagon.
Epinephrine in some organs. But glucagon is, you need the glucagon to break down the liver, in the liver, to get the glucose.
So in a fight or flight, insulin is decreased, glucagon is increased, which in turn increases your blood glucose level to give you more energy to fight.
Actually, insulin is increased too, because insulin allows the glucose into the cells. We'll go back, let's look. Stimulate torrent. Okay, what did he just wanted the glucose, didn't he? Insulin?
Insulin and glucose.
Okay, let's go back through it very quickly.
Oh, wait.
He just wants to know the effect of insulin release and glucagon release.
Okay. Beta cells, insulin, alpha cells, glucagon.
Yes. Yes.
Got it?
That's good.
Insulin is the main regulating hormone. Glucagon is supporting.
I think you might have wrote it wrong on the board.
But there are other hormones that regulate metabolism, and those are the like the epinephrine and norepinephrine and the cortisol and the aldosterone.
Yeah, they're minor.
Okay,
They're minor.
right. Okay.
Yeah, your mineralocorticoids, your glucocorticoids, your epinephrine in the fight and flight. Yeah, especially in the heart.
Okay.
Yeah, but that's minor compared to the insulin and the glucagon.
Understood. Thank you.
Insulin removes excess glucose, inserts GLUT4 into plasma membranes, skeletal, cardiac, muscle, liver, adipose. Promotes production and storage of glycogen, inhibits the breakdown of fat. Okay? Skeletal muscles, primary target, They take in the glucose for energy. The insulin, again, puts the GLUT4 receptors in there. Muscles take in the glucose and convert it to glycogen. It stays in there. Doesn't come back out. Energy storage, glucose is taken to the liver and skeletal muscles, converted into glycogen for storage. There's limited room, everything else then goes on as fat. Acts on the hypothalamus to inhibit the conversion of glycogen to glucose in the liver. So if the insulin is up, your liver is not going to be releasing glucose. So you're right, the insulin's going to be down in a fight or flight. When insulin level drops, the inhibition is lifted and the hypothalamus stimulates the conversion of glycogen to glucose to release it. Okay?
Okay.
So you've got potassium and calcium channels. Potassium closes, calcium channel opens, glucose is released. Glucagon, increased circulating glucose inhibits the alpha cells and glucagon is not released. Same thing as the insulin. If there's a lot of food, your body's not going to be releasing its stored food. It's stored energy. Glucose falls, then glucagon can be released. Prevents cellular uptake of glucose in skeletal muscle, liver, and adipose tissues. Promotes the conversion of glycogen to glucose and release from glucose from the liver. That's a glucagon.
So glucagon is just the stored form of glucose in the cell.
Liver.
No, that's glycogen.
Glycogen.
That's glycogen.
Glucagon allows the glycogen to be broken down into glucose and released from the liver. Insulin does not.
Yeah, I gotta get those.
Got that? Glucagon and glycogen. Glycogen is the storage. Glucagon releases it. What the muscle cells don't have,
Let me see.
yes,
What is it?
what the muscle, it's from the alpha cells of the islets in the pancreas. Okay?
Okay, say it again.
Alpha glucagon.
What do you want me to say again?
Okay, so glycogen is the storage or is it glucagon?
Glycogen
It's the storage. And glucagon is what?
is a hormone that releases it.
Okay, and then it releases it as a glucose.
Yes.
Okay, let me write that.
This is the glucose.
So if that's the
Glycogen.
It's inside the cell.
Is it released?
It's out the cell.
Well, it's the effect of glucagon. The action of glucagon, again, prevents the cellular uptake of glucose in the skeletal muscle, liver, and adipose. It's for the release. The glucagon is going to go up when the insulin goes down. The insulin goes up when the glucagon goes down. Promotes conversion of glycogen to glucose. Glucagon is when you need the energy. That allows the glucose to be burned, to be released. It doesn't allow the muscles to pick up and store it and make glycogen because they need the energy.
And insulin is the build level.
Is it what?
Insulin will be the build level.
Yeah, insulin would allow it to build up and store.
That's the burning we talk about. Burning.
Glucagon is the store, the burning, excuse me.
Got it.
Stimulates lipase and adipose tissue, converts stored triglycerides to fatty acids. The fatty acids are released into the blood. Remember what happens when a fatty acid goes into the Krebs cycle?
Yes.
You get about 108 ATP per molecule. You get a hell of a lot of energy.
Lots.
Okay? That's released by the glucagon.
That's the fatty acid you're talking about?
Yes, triglycerides generally. And they break them down into the because your triglyceride has 3 fatty acids. 16 molecules long, about 108 ATP per molecule.
That's why we use more fat. I mean, burn fat first when we're eating, right? That's where we get more of it.
Well, you start with glucose. You start with glucose, then you go to fat, then you go to protein.
Right. That's what.
Okay.
That's what. Okay. I'm just worried, but.
And aside, when you start a lot of physical exercise, very quickly you start with anaerobic glucose. Initially, it's anaerobic because you can put out a whole lot of energy very quickly, much faster than going through the Krebs cycle.
That's why moderate exercise burns more fat.
That's it. Moderate exercise will burn more fat than hard exercise.
Intention.
Hard thing to get across, especially to me. Metabolism while fasting. What's catabolism?
Breaking
Breaking
down.
Break it down.
You're taking your energy out, getting it out into the blood. Anabolism is building.
Building it up.
Amino acids, you need to know these. High
No.
protein, low carbohydrate. Gluconeogenesis or glyconeogenesis.
No.
Comparison, okay. Autonomic neurons. Activate while eating, stimulate the release of energy. Insulin, excuse me. Your body knows when you start eating, when you start swallowing, when your stomach starts filling up, your body knows that food's coming down and you are already releasing energy, insulin, before it gets there. Ready for that surge of blood sugar that's coming.
Oh, eating, I didn't know that.
Sympathetic division inhibits insulin secretion, stimulates glucagon. That's the answer to yours. You know, insulin goes down, glucagon goes up. Glucose cannot be stored if there's not insulin. Glucagon releases the glucose for the fight or flight syndrome. Okay?
So you said it cannot be stored?
If you have a fight or flight, your insulin's going to go down because you don't want to be pulling blood sugar out and storing it in the liver. You want it available to go to the muscles and the heart and the brain where it's needed. That's why your insulin goes down, your glucagon goes up because that releases a whole bunch of sugar.
So as soon as the glucagon goes down, the insulin goes back up.
Then it's starting to store. Sympathetic division inhibits insulin secretion, stimulates glucagon secretion, produces stress, hyperglycemia.
We don't need to know GIP and GLIP.
Glucose tolerance test,
Nope.
diabetes mellitus,
Yes.
chronic high glucose, inadequate secretion or action of insulin. If you're overweight, part of the problem is the insulin doesn't work. Doesn't mean it's not being secreted. You become insulin intolerant, or insulin tolerant, excuse me. You've got the insulin, it just doesn't work. That's inhibited by the fat cells, the PG something, PGA. Diabetes, miserable, major cause of kidney failure, limb amputation, blindness, heart disease, stroke. Type
And
1.
ED.
What's that?
ED too.
Wait, type 1 is juvenile onset?
Good to know.
ED.
Erectile dysfunction.
Okay.
Oh, yeah.
Check and see if he's drinking a lot of water at the same time. Okay, type 1 used to be called juvenile. Type 1 is probably an autoimmune problem. You just aren't making the insulin. The islet cells the islet beta cells of the islets are not, they've been attacked. We've changed that from juvenile onset to type 1 because we have so many obese juveniles now that juvenile onset could be a type 1 or a type 2. So, I mean, it's really kind of a
So
sad
then,
state.
if beta cells are destroyed, insulin will be replaced by...
It'd be an autoimmune. Injections of it, you have to
Right.
get exogenous insulin.
Oh,
It has to be injected.
okay, yes.
Okay?
But you need it, that's what
Juvenile, okay. Type 1 and fatty acids. Due to the reduced insulin, you're going to use more fatty acids for energy. You're going to see your ketone bodies go up. When you get out of whack, and we've talked about this before, you get what you call a diabetic ketoacidosis or a DKA. That is a life-threatening situation. You are so out of whack that you usually end up in the ICU, at least in the hospital, for a couple of days while they get your insulin in line and get everything back functioning, or get you back on an even keel.
So because reduced insulin secretion, does the more glucose in your blood and everywhere else?
Your glucose goes through the roof.
Yeah.
You eat a meal, your glucose goes through the roof because there's no insulin to take it into the cells.
And
or the insulin doesn't work.
It's not getting reabsorbed. It's not. I mean, it's getting sent out to your urinary tract.
It's in your urine. It builds up in the tissue. It becomes very toxic. You know, you lose your kidneys. You lose your. It breaks down your circulation. You lose your feet. You lose your arms. It's bad. Type 1, increased glucose, ketone bodies, excess dehydration. We talked about that. Why?
You're concerned about allow your body to reabsorb the water.
Yes, glucose in the proximal tubule does not destroys the osmotic gradient that pulls the water out. So it stays in the tubule, it's urinated out, you urinate a lot of water, and you dehydrate very quickly.
So that's why.
Glucose, type 1 diabetes have an abnormal high secretion of glucagon. Why? Because that's got glucose in there. If you've got glucagon in your muscles, it can be broken. It doesn't need insulin. If it's got in there, it can be broken down into glucose. Okay? It's using the glucagon to break down any glycogen you've got in for blood sugar, to get your blood sugar up. Enzyme converts fatty acids to ketone bodies. That's all part of the Krebs cycle. Insulin sensitivity. Obesity makes target cells less sensitive to insulin, so it doesn't work as well. Can lead to impairment of glucose tolerance or type 2 diabetes. Type 2 is impairment of glucose tolerance. Insulin independent. 95% don't need insulin, can be controlled by exercise, diet, and oral antiglycemics. Metformin is probably the most common one right now.
So since it says 95%, that shows that some people that are type 2s do use insulin shots.
About 5%. Yeah.
Because my dad did, and I used to work for a cardiologist, and the MA that trained me said, no, type 2s do not use.
They will, yeah. No,
But
some
maybe
will.
that
I've
was
seen-
just what he would say, so that, you know, I don't know, but
Well, that's why.
Okay.
yeah.
This is the same things that we've gone through, less severe. Triple to match the obesity rates. Two dead ways of mitigating type 2 diabetes: increasing caloric expenditures, lose fat cells that contribute to the insulin resistance, stimulates the production of GLUT4. Metabolic syndrome, is he going into that?
No, he's not.
Effects of type 2 we've already talked about. Hypoglycemia, that's when you can exaggerate insulin after a carbohydrate-rich meal. Reactive, limit your carbohydrates. Eat more sugar, eat more protein. Severe, can kill you. Thyroxine, growth hormone.
The adrenal hormones, yeah.
Adrenal, medulla, catecholamines, hang on. Glucocorticoids, thyroxine, thyroid. Now the thing you remember about hormones is they don't act on a cell unless the cell has a receptor. Thyroid hormone is put into the blood. It takes about 7 seconds to get to every cell in the body. The only cells that are going to respond to it are cells that have the thyroid receptor. Increased production of metabolic heat. They thought they could use thyroid for diet to raise your metabolic rate. It didn't work real well. Acts on most cells in the body, increased rates of cellular respiration, decreasing intracellular ATP. BMR is basic metabolic rate, independent of thyroxine or dependent. Thyroxine down is needed for proper growth and development of the CNS. needed for balance between anabolic and catabolic process.
Where do we need thyroxine?
It's the brown fat cells.
Yeah, part of it is, but it's just, it regulates the body metabolic rate.
I think the connection he wanted us to make was with brown fat cells.
I wish you'd said that.
What?
Did Sarah go over this in Asana on Friday?
She didn't mention brown
We skipped
fat cells.
thyroxine because the only thing that she thought that he was going to want to know
So what?
on
He wants us to.
brown fat was the proton gradient,
Right.
and that's why it's
And
constantly
coupling.
working, and that's why there's so much heat.
Remember how they said adaptive thermogenesis, which is the opposite of shivering thermogenesis, which comes from our brown fat cells?
Yeah, the non-shivering thermogens.
Yeah.
And that's
Brown
our
fat.
brown fat cells. So thyroxine is what causes that protein to be in the mitochondria of the brown fat cells, so they produce more, increasing our metabolic rate.
Okay?
Okay, good call, Matt.
So what brown fat cells normally then?
There's very few in the adult. They're in the neck area. They're mostly in the babies because the babies don't have the fat and the ability to control their body.
So they're taken from there and carried to the cells that need it?
No, it's where they're broken down. They're broken down there and it just releases heat. Okay, CNS, growth hormone, no. What hormones was he looking at?
Just the
Thyroxine?
thyroxine, the corticoid, the mineral corticoid, the glucocorticoid, and the norepinephrine and the epinephrine. Those are the 5 I got. Those guys, yeah. The catecholamine.
Catecholamines?
Yeah, that's the norepinephrine and the epinephrine.
Catecholamines are releasing glucose from the liver.
Yeah.
I don't have anything that says that
Yeah,
in my stuff.
All right.
Glucagon and epinephrine.
No.
It's a release of corticoids, glucocorticoids.
No, I don't know.
Glucocorticoids, cortisol, general adaptation in response to stress.
Yes. It's
Yeah,
actually
you need
there.
to know that.
Yeah, it's there. It's under chapter 19, underneath what triggers glucagon release, what are some other hormones. It's the last 2 slides. But yeah.
It says what triggers it. Not glucose.
Okay, you just need to go back and look at it then.
Yes.
If you're
On
burning
your
out on it,
reproduction.
Yeah.
you need to go back and look at it.
Okay. Let's go to reproduction.
Calcium and phosphate.
No,
vitamin D,
skip it.
parathyroid, calcitonin.
Nope.
We already did that earlier on, sort of.
Nope. Reproduction.
Woo-hoo.
Sexual reproduction.
Do it.
Genes, random ways to produce a new individual, 2 genes from 2 individuals. Germ cells become gametes in the gonads. You think it asks the difference between gametes and gonads?
He got us.
Yes.
Gametes is, are the cells, gonads are where they're produced.
Your cells become candidates.
What? Gametes of what?
Sperm and ova, the cells.
Doesn't it become an ova when the sperm fuses with it?
It goes through the final
I thought it
meiosis.
was the secondary oocyte before the sperm.
Yeah.
And so when the sperm drops the nuclear stuff off, it becomes the ova, and then it does its
does
next
one-
meiotic.
It does one more meiotic, and then the other one disintegrates.
So when the sperm enters the nucleus, the sperm.
Because when the sperm enters the egg, it finishes its meiotic division.
So is it ova when the sperm enters before meiosis, or ova after the second meiotic division?
It's ova when the ova is the egg that comes down out of the ovaries. It's an ova at that point.
Wow.
Yeah.
Because after it fuses, it's a zygote.
This is like this. Yeah. It's a zygote. And
Okay.
it's an embryo.
Germ cells become gametes, sperm and ova in the gonads via meiosis. They're in the gonads. It's in the ovary and the testicles.
Right.
So it's an ova at that point. When it's fertilized,
So
it's a
are
zygote.
ova and a secondary oocyte the same thing?
Well, you know where you have your oocyte, and then it goes through the first meiotic division. You have a secondary oocyte, which is released. It's still an ova.
So is it an ova when it's released from the ovary?
Yes.
When it's going down the fallopian?
Yes.
That's now an ova.
That's an ova.
Okay.
And the oocyte
Got it.
before that, I think.
Yes.
Yeah, I think
Secondary
oocyte.
oocyte's what gets released from the graphian and pollen.
Sexual reproduction.
So on this one, adult male, you have the sperm, you've got the ovum,
Okay,
and the zygote is after they've been fertilized.
yes.
23 chromosomes from mom and dad. Females have 2 X. Mom passes on an X. Males have an X and a Y. The sex is determined by the male.
Horrible.
It's the last decision we get to make. X and Y look different, have different genes. Y has many testes-specific genes. One X in the female is active, that produces the bar body. We talked about that when we did the simple stain. Sometimes on the neutrophils and the cells you can see that bar body. They say here, an easy way to visually determine the sex of a cell. That's not really true. There's a picture of your bar body on the right.
Cool, bar body.
Formation of gonads. As the child is developing, for about the first 6 weeks, it could be either male or female. You have, from the Y chromosome, you have the Leydig cells, or testes or ovary signaling determines, is called testes determining factor. It's coded by the gene of the Y. When the testosterone is produced, it produces testes. If the testosterone is not produced, the testes go away, and you have a female.
Which is estrogen. Right?
He talked about SRY.
What's that?
He talked about SRY.
It's just the name.
Yeah, that is the TDF.
Well, yeah, that's what I read it in the book.
You mean
It's the
the
exact
SRY?
same thing. But
I think the TDF was before they found the SRY. Isn't that what they said?
SPY is what determines a male.
I'm going to have to look. I don't.
Like, is it catch-all?
That could be. TDF in XY embryos, seminiferous tubules form, germinal cells, Sertoli cells, Leydig cells make testosterone. Leydig cells are the cells throughout the male's life that are going to make testosterone. They're very active.
Oh, the lady cells?
Well, the embryo is growing, then they go essentially dormant until puberty. Begin to make large amount of testosterone at 8 weeks. As they develop, the testes descend into scrotum. After they're all developed, it pretty much goes away for a while. Without the TDF, XX embryos do not develop testes. Follicular cells do not appear until the second trimester.
Well, that's a good one to put on our notes.
yeah.
Accessory sex organs.
Skip.
Oh, doesn't he? He has it on mine, doesn't he?
He has accessory male organs, but he doesn't have,
Where's he at?
Sex determination,
in what trade?
male and female reproductive systems developing a
Those
reproductive organ,
highlighted ones?
role of FSH and knowledge.
Oh, yeah.
That's what
I know this because I think it's cool.
So how sex is determined, I thought that the answer to that was the SRY gene.
Yeah, that is
Wolfian.
Well, it's the X to the Y. I'm not sure what you're saying with SRY. I'm not sure.
SRY is the same as TDF, according to the textbook.
Okay, let's see what we get to here. You have the Sertoli cells, Wolfian ducts become the tracts, Mullerian ducts become the female tracts. Men have an anti-Mullerian hormone that's released, so it inhibits the female tracts being formed in the men. It's a Mullerian inhibiting factor secreted from the Sertoli cells. Without this, a man will develop fallopian tubes in a uterus. They won't be workable, but
Wow.
That would be funny.
external genitalia after 6 weeks, testosterone, masculinize it. With lack of testosterone, you form the female genitalia.
That's a good picture of Brian knows.
Sure.
You gonna draw that?
Here, Patrick. This is what it'll look like.
Okay, there's your timetable. This is all in your book. Sexual disorders, hermaphrodism.
So working cells drop both female males?
What's that?
That's dropping both?
If you don't have the anti-malarian factor, you'll develop the duct. You'll develop the ovaries.
What did you say makes the anti-malarial?
Sertoli cells.
That's what I thought, okay, thanks.
So the Wolffian develops them both up until the other one inhibits the malarian, right?
No.
It shows a different time frame, that's why I was asking.
Wolffian ducts become the male tract.
But they also,
Malarian ducts become the female tract. So they're developing young. But then you have the anti-malarian factor from the Sertoli cells that keep the female tracts from developing in the male.
wait, Sertoli cells do what?
They excrete the anti-malarian inhibiting factor,
Okay,
anti-malarian.
all we need to know about Sertoli cells is they feed and protect sperm by isolating them to the blood system because of the autoimmune response.
Okay, then
I
you
thought
got
it was an interesting fact. Let's move on.
external genitalia. You have testosterone, you produce it. You don't have testosterone, you don't. Testosterone, you produce male. No testosterone, it's female. You don't need to go through this disorders.
No. Endocrine regulation.
No, I don't think you're going into that at all.
No, I thought we would.
I mean, by this time of the semester, he's just trying to get through.
Yeah, they didn't even talk about conception. No.
Okay. Testes stop, like I said, making testosterone very important. Narrow ovaries don't even start until puberty.
So both until puberty, right?
What's that?
Both until puberty, both
Yep.
female and there.
And at that time, your anterior pituitary is making gonadotropic hormones. FSH and luteinizing hormone are produced in both males and females, stimulate spermatogenesis or oogenesis. Okay? Stimulate gonadal hormone secretion. Maintain the structures. Release of FSH and LH is controlled by gonadotropin hormone releasing hormone from the hypothalamus, regulated by negative feedback. I assume he may be asking you about negative feedback.
Yes.
Aside from the gonadal hormones, testosterone, estrogen, they secrete inhibin. Inhibin by the Sertoli cells or granulosa cells in the ovarian.
Can you
It
go
inhibits the release of FSH.
back? Mr. Smith, can you go one back, please? Thank
Right
you.
there. Let's see. This negative feedback is on the ovulation section, right, Mr. Smith?
It's in chapter 20, yeah. One point that they want to make here is the testosterone is being made constantly. The LSH, FH, the female hormones are cyclic in the female, which accounts for your period for one thing, and it keeps them from becoming sensitized. So, you have these spurts of FSH and LH, and then they go away, so the cells aren't sensitized as if they're ready to respond again with the next spurt.
They're responding.
But if they're exposed constantly, we talked about sensitization, it happens with hormones, and they, after a while, they just stop responding. So the hormones in female are what they call periodical. Okay, cyclical, how's that?
That's cool.
Okay, female cycle, estrogen has a positive effect on LH release. LH goes up, estrogen goes down. Puberty, FSH, LH are elevated. And then they decline until puberty.
And then they go crazy.
And it goes crazy at puberty. Development of increases in testosterone and estrogen, and then secondary sex characteristics come out after the hormones are released. In the male, the voice deepening, the enlargement of the penis, and things like that. Female, breast, menses, pubic hair on both male and female. Breast, growth spurt, breast development, menstrual on female. Body, muscle, penis, and testes growth on male. Hair on both. Depends on the levels and amount of body fat.
Oh, yeah.
Mr. Smith, LH is the only hormone that stimulates secondary characteristics, right?
No.
It's both?
No.
FSH and...
Well, LH stimulates the testosterone and the estrogen. The testosterone and the estrogen stimulate the secondary characteristics.
So FSH is not involved?
I don't believe so.
So
No.
LH...
Okay. LH triggers the testosterone and
In onset of puberty, especially in girls, the thinner, the less body fat, generally the later the puberty. They found like in World War II, women that were fighting in the underground, high stress, low body fat, they were always near starving. They stopped having periods, and then as soon as everything stopped, they went back to a normal menses and things like that. Okay, sexual response, did you get into that?
yeah. We did need to go through this again.
And it was just, never mind. Male.
I think there was something about knowing that men can get excited more than one way. That was something we didn't know.
Oh, yeah.
They get
We know.
excited by brain and by touch. I think it about
Visual, touch,
Right.
yeah.
Telephone.
Excited by brain and touch, too.
Rose, we don't need to know about your second job.
That's good.
And hopefully everybody will forget that by next semester. Okay, testes. Are we in? Did he get in? You need.
Yeah, testes, yes we do.
Oh,
Lots of testes.
Reproductive organs. Okay, let's go through this very quickly. We're almost through.
Oh, is it the matching?
Yeah.
Oh, okay.
Structures and function matching only for reproductive system.
Well, you have something that I don't.
No, Patrick told us.
Okay. Because I just got what he sent.
He verbally said it.
I write down what he says.
Okay. So, 2 compartments, seminiferous tubules, where it spermatogenesis, where the sperm are formed. And then you have
Oh, right there.
seminiferous tubules, FSH receptors, Sertoli cells, spermatogenesis. Interstitial tissue, Leydig cells make testosterone, are filled with blood and lymphatic capillaries. LH receptors found in the Leydig cells cause the Leydig cells, LH cause the Leydig cells to secrete testosterone. LH secretion is controlled by rising testosterone, negative feedback. FSH secretion is controlled by testosterone and inhibit is from the Sertoli cells.
So the luteinizing hormone for males,
They both, males and females, have LH and FSH.
But for males, the LH, what does that do? I know for females,
It causes the secretion of testosterone.
oh, so that's a feedback right there, right?
Testosterone in the brain. There's probably two big views of this, the male view and the female view. But converted by 5' alpha reductase to estriol, and estriol inhibits LH. That's your negative feedback.
But he talked about it as estrogen in the ovarian and the menstrual cycles.
Yeah, well, that's different. Spermatogenesis, diploid, go through mitosis, one daughter cell. After meiosis, you have two secondary, meiosis 2, 4 spermatids. And you've got one goes to 4. Spermiogenesis, maturation into functioning, usually takes place in the epididymis. Sperm redevelopment requires Sertoli cells, create a blood testes barrier. This is actually very important. Sperm is considered by the body as foreign. The Sertoli cells make the testes immunologically privileged, meaning that the body can't get in there and look and see that these are foreign. What happens after a vasectomy sometimes, when the spermatocords are cut, you will sometimes get sperm leakage into the body, and then you will develop the anti-sperm antibodies.
That's not good.
Well, you're not fertile anymore anyway.
Oh, so that
The
way-
problem is if the female develops an anti-sperm antibody. And then they're essentially infertile at that point.
How would that happen?
What's that?
How would that happen?
Her body's just able to recognize.
It's just at some point, and it's very rare, the female body recognizes
No, I love my kids.
the sperm as foreign before it can get in and fertilize the egg.
Oh, is it?
And so it attacks it much more vigorously, and the chances of getting pregnant are very.
But it would be that one particular sperm because it doesn't have every sperm its own HC whatever. Its own.
It has their own MHC.
Well, yeah, but normally a female does not have an anti-sperm antibody. So.
Crazy.
Okay. Sertoli cells envelop developing sperm. Concentrates the spermatozoa in the seminiferous tubule. Let's see here. Testosterone stimulates meiosis. FSH enhances it. And spermatids move through the seminiferous tubules to the rete testae, efferent ductules, and the epididymis. Epididymis is where they mature and store. Ejaculation, they move from the epididymis to the ductus deferens, ejaculatory duct, and the urethra.
Can you leave that slide just for 1 minute?
Sure.
See that, please?
Now, when you on the lab, didn't they go through that in the movie?
Yeah,
On your movie lab?
the miracle
Yeah,
blood.
they showed it
Miracle
all.
life.
Yeah. But I took a lot of notes, but I didn't.
It went a little bit fast.
Yeah.
It's just like it goes from here to here to here.
Yeah, I kind of went.
And it's like, okay.
Okay. You guys got it?
Yeah. Thank you.
Okay. Prostate, seminal vesicle prostate gland add fluid. Seminal fluid is full of fructose for energy. Prostate is citric acid, calcium coagulation proteins. Sperm, after it's ejaculated into the vagina, coagulates for about 20 minutes. Then it liquefies again. When we do a semen analysis, hopefully the specimen has been collected in the lab and we can measure, see that it does coagulate and that it does liquefy after that. That's something we would report to the doctor. Erection, blood flow into the penis, parasympathetic nervous system induces vasodilation. Nitric oxide is a neurotransmitter. Any questions on that? Control by the hypothalamus, sacral region of the spinal cord. Can occur due to conscious sexual thought or sensory stimulation. You say this is all part of the matching?
Yeah.
Answers on? Okay. Emission is the movement of the semen. Ejaculation is the expulsion into the vagina.
That's kind of confusing, so both under sympathetic.
Ejaculation.
But it's not about, well, yeah, parasympathetic, it just goes in order. Parasympathetic leads to.
Sympathetic is
Sympathetic is the actual process of ejaculation.
Right, you have to contract the muscles, right?
You can't control
The
it.
erection is relaxation.
You can't control
The
it.
parasympathetic.
Sperm count, 20 million per mil. Vasectomy, they cut the vas deferens.
Is that the best estimate right there, that photo?
What's that?
Is that the best? Yeah, it is.
I don't know.
Is that where the leakage might happen?
Yeah,
Okay.
yeah.
Doesn't look like a bastard.
Usually they tie it off and usually they cauterize it too.
Look at, they just
And
kind of.
sometimes they grow back together. Sometimes they will connect. And every once in a while, a lady
They just tie them.
whose husband, they tie them and clip them. They clip a segment out.
She gets pregnant anyways.
She gets pregnant anyway,
Yeah.
which causes a whole lot of marital problems.
Why
Uh-huh.
wouldn't they,
Yes,
why would
it
they
does.
laser it, like burn it or laser it?
What's that?
Why wouldn't they burn it or laser it? You know?
They do.
They do, okay.
They do. They cauterize.
I thought it was just tied on, I wonder if it's leakage.
Female reproductive. Okay, let's hold it together for another 10 or 15 minutes and we'll be done.
Okay.
Ovaries, gonads, oocyte, and sex steroid production. Fallopian tubes have the fimbriae or the cilia that sweep the egg along. Uterus, site of embryonic development. Endometrium, implants, myocardium, muscle layer, contract, expel, peritoneum, connective tissue. Cervix, the narrow region at the bottom of the uterus. Vagina. Ovarian cycle. Toward the end of gestation of a female's oogynia, begin meiosis, produce primary oocyte. Ovaries of a newborn girl have about 2 million. By puberty, they're down to about 400,000. And 400 of these, about 400 will be ovulated. So, one a month from 13 to 50, something like that.
That's a lot.
The thing about it is, if a lady gets pregnant at 50, her eggs are 50 years old. Anything after about 35, you're really looking very closely for Down syndrome and things like that, because the eggs are just getting too old. The male, on the other hand, are producing new sperm constantly, pretty much for their life.
Yeah, but I just saw in 60 Minutes that a male who was like 40 years or older and like has a kid, like his sperm, you know, when he's 40, has a kid, there's more chances of the kid having like autism
Oh, wow, really?
at the age of 40 or older.
Oh, they get it, they notice it at that age, right?
Yes, well, the rates of autism throughout the entire population are going up
That's due
tremendously.
to estrogen on C.
It's all the sperm.
And I don't think, that may be part of it, but I'm pretty sure that's not the whole story there. Going on, FSH, some of the primary follicles grow to reproduce. Granulosa cells, undeveloped fluid-filled vesicles, secondary follicles.
You're so sweet.
These secondary follicles fuse vesicles to form a single antrum. This makes a mature Graafian follicle.
I don't remember.
Cell layers are listed below. Corona radius,
Can't
zona
test
blue.
for it?
List it up.
You remember two specific?
Okay, so you've got this started. Now it takes one Graafian follicle. Occurs because of the FSH. Paracrine regulators such as androgens and FSH induce apoptosis of the other. What's apoptosis?
Cell death.
Cell death. The others die. The one that doesn't die grows to a primary oocyte, become a secondary oocyte, plus a polar body which degenerates. Secondary oocyte finishes meiosis only if it's fertilized.
You go back to the picture?
Wait, secondary oocyte finish my own. It sounds
Oh.
The Graafian follicle grows, the primary oocyte finishes meiosis. So of all the Graafian follicles, all of them die except one. The primary oocyte finishes meiosis to become a secondary oocyte.
So I have to go to meiosis 1 to become a secondary.
I learned something interesting today about the secondary and the primary. The secondary has 23 chromosomes. Is that correct?
Yeah.
Yeah, and then the primary has 40.
46.
Yeah, so that's the first time. If you hadn't told me, I would never known that.
So 46 and 23?
But
I mean, it makes sense.
It doesn't
It's
finish the
very
meiosis.
developed.
Secondary doesn't finish the meiosis until it's
Right.
fertilized.
I think that was the take home was that it's not the same as spermatogenesis. Spermatogenesis
No.
finishes the second meiotic.
Spermies are down to 23 when they start the journey.
The primary has the 46.
Yeah.
Where the egg
Even the secondary has the 46 until it's
See, now that's what I
fertilized.
thought. That's one of the, okay.
Even the secondary.
Then it
has-
finishes meiosis, you get a polar body which degenerates.
Oh, doesn't make it.
But eventually.
So the other 23 that are broken off into meiosis form the polar body that degenerates. But it's after fertilization.
That's the
That's
master
what
ones with
it
the,
sounds.
that's the master ones with the
Wow.
sperm on. Hold on.
I don't know exactly how that happens. Ovarian cycle, stimulating development of follicles. FSH stimulates estrogen production. The bigger the follicle, the more estrogen it releases. Surviving Graafian follicle becomes so big it bulges out of the ovary. It bursts and releases the secondary oocyte. So it develops in the ovary, it pushes its way through the membrane and then bursts and releases the oocyte to move down the fallopian tube.
Dylan mentioned this earlier, he's not here, but if we ovulate every month, right, and we don't have a baby, that's a scar from the corpus luteum turns into a scar tissue. Right.
Yep.
Now, it seems like they just get bigger and bigger, something like a run out of room, because the way the picture is, they're pretty big. Like,
The luteums.
does that make, it's just, I don't know, it's hard to see.
Well, you do see that. You see that in,
I know that
in some
people
you know,
don't
some-
ovulate every month too, but still, even if every six ovulations out of the year or something in our adult life, that's a lot of scar tissue in your ovary.
about 400 little bits of scar tissue, yeah.
Is that why maybe some women have problems, no?
Bye, man.
Have a good one. Good luck on your test.
You need it. You're gonna get 100.
Well, that's why fertility goes down.
Say it like that, wait.
It was hard to picture it, like, all this scar tissue, doesn't it cause problems?
Well, your graphene follicles moving to the edge of the ovary, and then it's bursting through the ovary, and then, yeah, you're developing scar
Is
tissue.
that why you shouldn't have kids up until a certain age, because you have more scar tissue by then? I mean, could that
Not
be the
age.
problem? You know, when you're 50, you shouldn't really try for kids?
Well, the thing about 50 and trying for kids is your chances of a
Yeah.
mutation,
My-
trisomy, Down syndrome, and that go through the roof. I mean, from the time you go to 35 to 50, it's probably, I'm trying to remember, I think it's like 10 times more chance you're going to have some sort of mutation, just because of the age of the eggs. It's tremendous.
So you should have an amniocentesis then.
Oh, it's required. If you're over 35 and you have, and you get pregnant, they're going to do an amnio.
Like they usually do a blood test and then they decide.
They do, well, no,
There
they
wouldn't
do
be a deciding.
an AFP,
You got all these.
but if you're over 35, they're going to do
They
an
have
amnio.
me do it no matter
Yeah,
what,
it's,
even if you don't want
the
to.
AFP is just not that
First
simple.
trimester now, too.
Um, I'm not familiar with that. Okay? One, good luck, Rose.
Your cake is yummy.
Okay, after ovulation, the follicle becomes a corpus luteum, secretes both estrogen and progesterone. Hormones play in the cycle, the menstrual cycle. Do we need to go through the menstrual cycle?
No.
Cyclic changes.
These are the important points, but I don't feel like reading. No, that's
That's
gross.
It's kind of big.
Yeah.
Yeah.
That scar tissue is different. Is that the actual hole? That's the corpus luteum.
How did they get that is what I'd like to know.
What's that?
How did they take
Oh, they
that?
run small cameras up. I mean,
Oh, okay.
look
but
at that
when
Miracle
they
of
do a
Life, the small
C-section or
cameras
something,
that they
that's
have.
Yeah, it's still pretty big, right?
Yeah.
Have you seen Look Who's Talking? Oh, I know, right? I love that.
You know, that was actually pretty correct.
Yes.
You know.
It's such a cute movie, too. Yeah. Oh my gosh, that's adorable.
So we don't need to really go through the
No.
cycle, female cycle. And I think that's it, guys.
Cool.
Yeah. And he didn't go through that.
Contraception, menopause, fertilization, he didn't get to, right? So he's not testing you on it?
Well, we just need to know that where it occurs, and it's in the fallopian tube, from what I understood.
Right.
It's not in the uterus, it actually happens in the fallopian tube.
And then it moves to the uterus and implants.
Yes.
Yes.
Or we also need to know that once fertilization occurs, that the corpus luteum makes the estrogen and progesterone to sustain the implantation until the placenta develops enough to start regulating itself.
Right, that's coming from the corpus luteum.
That's, I think, I didn't get anything else on that. No, that's it.
And then we're into the cleavage and the blastocyst, and I guess you didn't get into that.
No, we did that last, that's like we did.
Sorry.
We didn't talk about the
And
placenta, right?
Amniotic sac.
We talked more about the placenta and the anatomy of that process. He goes straight to the reproductive system. He loves the physiology on that.
Well, you know, if I had to prioritize it, you know, what, if you're going into nursing, you know, you look at the endocrine system, you look at the immunological, you look at the renal, you look at the reproductive, and the circulatory, respiratory and circulatory. So you have 7 or 8 chapters out of the 20 that we went through that, as a nurse, specifically, you know, something that you would use every day. The rest of them, if you're going to medical school, yes. But
But they're going to take more advanced classes too.
yeah, they're going to get stuck with this again.
Many times.
Well, yeah, and they're going to break it down. You're going to have, you know,
One class on reproduction.
Yeah, right.
One class on renal, one class, a quarter or semester, and they're really gonna get into it.
I'm taking pathophysiology next semester.
Here or where?
In Alabama.
Yeah, they passed on a pathophys book to me.
Mr. Smith, are you downloading this on YouTube?
If I get a chance, I'm sure I'll have it on by Thursday.
Okay.
You know, you
Wait a
should
minute,
What makes troponin move is calcium.
Why don't you just get an iron site? That'd make it so much easier for you.
Get a what?
We already have iron.
Like, is the blood
That's something like that's where we go for online classes or for the like.
That's for the school, isn't it?
Yeah, but my lecture classes, all my teachers use
said
it.
the word a different way, right?
Like, huh?
Iron.
I don't know anything about it, to be perfectly frank with you.
Well, then I'm just saying, because you are.